Welcome to the Deep Dive, where we sift through the most complex scientific research you send us and extract the knowledge you need to be truly well-informed. Today, we are undertaking a deep dive into rabies, a pathogen that is both ancient and, paradoxically, a cutting-edge example of evolutionary mastery. When you think of rabies, you probably conjure up vivid, terrifying images. An animal displaying aggression, perhaps, foaming at the mouth. But what our stack of sources reveals is that the virus causing this horrific disease, the Lyssavirus genus, is not some crude brute force killer. It is, well, a molecular genius, really, a masterclass in viral efficiency and profound stealth, orchestrating a progressive, fatal acute encephalomyelitis. Our mission today is to pull apart the virology, the incredibly compact genomics, and the cutting-edge global tracking of this highly neurotropic agent. We want to understand how it operates, how it systematically dismantles the immune system with just 5 genes, and what the latest breakthroughs in cryo-EM and genomic epidemiology mean for the global goal of eliminating this disease.
And that tension is where the real story lies, isn't it? You have a virus with an instruction manual so small, just those 5 genes, yet it is nearly 100% lethal once symptoms manifest. To appreciate its efficiency is to, understand why rabies has been so difficult to fight for centuries. We have to drill down into this precise molecular architecture because it's the only way to refine our existing countermeasures, whether those are vaccines or immune globulins, so they remain effective even as this virus evolves quietly in wildlife reservoirs.
So before we get into its structure, let's place the virus on the evolutionary map. We know it's a Lyssavirus, but where does that fit into the grand scheme of pathogens?
It occupies a very specific niche. The agent belongs to the genus Lyssavirus, which sits within the family Rabies. This family is characterized by those distinctive bullet-shaped particles.
Oh, right.
But the larger order is Mononegavirales. The name is the clue mono because they have a single non-segmented RNA genome, and negative because that RNA is negative sense, meaning it can't be translated immediately by the host cell.
Yeah.
The genus name itself, Lyssavirus, is derived from the Greek goddess of rage, Lyssa, a fitting, if grim, tribute to the disease's manifestation,
Yeah, definitely fitting. So how diverse is this lineage? It feels like we mainly hear about the classic rabies virus, but I understand the family tree is cool.
Oh, it's far more diverse than the public realizes. The International Committee on Taxonomy of Viruses, the ICTV, currently recognizes a complex family of at least 17 distinct Lyssavirus species. And there is a striking ecological feature that unites them. Bats, the order Chiroptera, are considered the ancestral and primary reservoir hosts for almost every one of these species.
Almost every one.
Yeah, there are only rare exceptions, like the Mokola Lyssavirus, or MOKV, and Icoma Lyssavirus, or ICOV, where we haven't definitively confirmed a reservoir host cycle yet. But mostly it's bats. This diversity necessitates organizing them into phylogroups for, public health tracking and understanding risk.
Right. And naturally, Phylogroup first is the one that causes the overwhelming burden of human disease, I assume.
Exactly. Phylogroup I is the most clinically significant. It contains the classical rabies Lyssavirus, or RABV, which accounts for well over 99% of the tens of thousands of human deaths globally each year. This group also includes other serious threats like the Australian bat Lyssavirus, ABLV, and the European bat Lyssaviruses, EBLV1 and EBLV2.
And where do they circulate?
Well, RABV maintains transmission cycles in carnivores worldwide and bats in the Americas, while the other Phylogroup I members are mostly contained within Old World bat populations.
Okay, that leaves Phylogroups the second and 3. If they aren't currently causing widespread human death, why should they matter to us? Are they a future threat?
They matter profoundly because of what we call the global vaccine gap. Phylogroup II, primarily African, includes viruses like Logos bat Lyssavirus, LBV, and MOKV. Phylogroup III contains the unique West Caucasian bat Lyssavirus, WCBV. Our sources highlight a critical vulnerability related to these sort of more distant relatives.
Vulnerability. What are the real-world consequences of that genetic distance you mentioned?
The problem is immunization. See, while there is strong serological cross-neutralization within a phylogroup, meaning if you are vaccinated against RABV, you are largely protected against ABLV, for instance.
Okay, that makes sense.
There is extremely limited to virtually no cross-neutralization between phylogroups. All current human and veterinary vaccines are derived from and optimized against Phylogroup I RABV antigens.
Ooh.
Right. Now consider the implication. Phylogroups the second and three are circulating silently in bat populations. If there were to be a significant spillover event, say a bat Lyssavirus from Phylogroup II action jumping into a domestic animal population,
then our standard treatments might fail.
Exactly. Our current standard post-exposure prophylaxis, or PP regimes, would likely be rendered ineffective. This vast, silent genetic diversity circulating in bats represents a latent public health threat that requires us to rapidly develop broadly protective vaccines.
Wow, that reframes the context entirely.
Yeah.
Knowing the stakes, let's look at the structure that must evade these potential new vaccine challenges. That iconic bullet shape is instantly recognizable, isn't it?
It is perhaps the most defining feature in the Rabies family. The RABV virion is an elongated cylindrical particle, typically about 180 nanometers long and 75 nanometers in diameter. It's always structured with one conical, sort of rounded end and one planar flat end. Think of it as a precisely engineered two-layer capsule.
Okay, let's detail those layers. On the outside, you have the host-derived envelope, right? Stolen goods, essentially.
Correct. The outer layer is a lipid bilayer that the virus essentially steals from the host cell as it buds off. This envelope is studded with approximately 400 spike-like projections. These spikes are trimers of the viral glycoprotein, or G protein. The G protein is the key, master of ceremonies for infection. It mediates attachment to the host cell receptors, facilitates fusion for entry, and is, critically, the sole antigen targeted by our neutralizing antibodies.
So if you get a rabies vaccine, you are training your immune system against this G protein specifically.
Precisely, that's the target.
Now, beneath that vital outer layer, you have the scaffolding that holds the shape together.
That's the matrix, or M protein, which forms a dense layer just underneath the envelope. The M protein is the architect, you could say, the construction manager. It links the external envelope to the internal core, orchestrates the entire assembly process, and is fundamental to imparting that characteristic bullet shape.
And deep inside, you find the cargo.
Exactly. The RNP core, the ribonucleoprotein package. This is the helical nucleocapsid containing the precious single-stranded RNA genome tightly coated by thousands of copies of the nucleoprotein N, along with its partners. The phosphoprotein P, which is an essential cofactor, and the huge, large protein L. That's the catalytic RNA-dependent RNA polymerase, or RDRP.
I find the latest structural insights so compelling, especially what cryo-electron tomography, or cryo-ET, has told us about the virus's movement. It suggests the bullet isn't a rigid shell, but something more.
Well, dynamic. That's right. The classical textbook image often shows a fairly rigid structure, but cryo-ET has revealed that RABV exhibits significant structural heterogeneity and pleomorphism. It's flexible, it can bend. This fascinating difference is traceable to the matrix protein's stoichiometry, specifically the ratio of M protein to N protein.
How does that ratio change the structure?
Well, if you look at a close relative, the vesicular stomatitis virus, VSV, which is known for its rigidity, it actually has two distinct layers of M protein, giving a 2.1 MN ratio. This dual layer acts like tight internal bracing, locking the RNP helix in place.
Okay.
RABV, however, has evolved to use only a single layer of M protein, resulting in a 1.1 MN ratio.
So the reduced scaffolding allows for movement. But is that flexibility a risk, or is it an advantage for the virus? Seems like it could make it weaker.
Our sources suggest it's actually a profound adaptive advantage. That reduced M content allows the internal RNP helix to flex more freely. This inherent maneuverability likely enhances its stability across different environments.
Like what?
Like surviving in highly variable saliva outside the host, perhaps, or navigating the incredibly crowded, complex intercellular spaces within the central nervous system. In the dense environment of the CNS, being able to flex and bend could ease its passage, enabling rapid and efficient neuroinvasion. It seems built for operational necessity, prioritizing efficiency over rigidity.
That is a brilliant piece of engineering on the outside. Now, let's pivot to the instructions inside that RNP core. You mentioned the stunning genomic economy, only 5 genes. How does this virus maximize functionality from such a, well, minimalist blueprint?
It is the ultimate lesson in efficiency, really. The RABV genome is a linear, non-segmented, negative-sense, single-stranded RNA molecule, a mere 11 to 12 kilobases long. Because it's negative-sense, it's useless to host ribosomes until it's copied into a positive-sense messenger RNA, or mRNA. The entire system is built around controlling the output of its 5 genes, and the gene order is the absolute key, 3-NPM-GL5.
Why is that specific conserved order so crucial? What does it do?
Because it dictates a self-regulating system called the polar transcriptional gradient, or the stop-start strategy. Think of the genome like an assembly line for protein production. The viral polymerase, the L protein, can only initiate transcription at the very beginning, the 3' end.
Okay.
And then it proceeds sequentially down the line, N through L.
And what happens at the junctions between the genes? Does it just read straight through?
No, that's the clever part. At each short, non-transcribed intergenic region, or IGR, the polymerase terminates the previous gene's transcription. Then, when it attempts to reinitiate for the next gene, for example, moving from N to P, it's highly inefficient. There's a high probability, a calculated chance, that the polymerase will simply fall off the template entirely.
That sounds counterproductive, like it's designed to fail. But you're saying it produces exactly the distribution the virus needs.
It is brilliant, actually, and it prevents the need for complex, host-dependent feedback loops that could be easily disrupted. Because of this fall-off probability, the gene closest to the start, N, is transcribed at the highest level, P is next highest, and M and G follow, with the L gene way at the distal end, transcribed at the absolute lowest level. This gradient N, P, M, G, L perfectly matches the virus's stoichiometric requirements. Thousands of N proteins for encapsulation, fewer M and G proteins for structure, and only small catalytic amounts of the huge L polymerase needed.
That hardwired genetic economy ensures survival. It's incredible. Now, where does this hyper-efficient transcription actually take place? We rely on finding the pathognomonic Negri bodies for diagnosis, which used to be seen as simple junk piles inside the cell.
Yeah, they are anything but junk piles. Negri bodies are the signature hallmark of infection, absolutely essential for diagnosis historically. But modern virology has revolutionized our understanding. We now classify them as dynamic, liquid-like organelles known as biomolecular condensates.
Liquid organelles. That's a huge shift from thinking of them as static inclusions.
Mm-hmm.
How does a liquid organelle form spontaneously inside a cell like that?
It comes down to two of our viral proteins, N and P. Both contain what are called intrinsically disordered regions, IDRs. These are flexible, sort of spaghetti-like domains within the protein structure. These IDRs mediate weak yet multivalent interactions. When these proteins hit a critical concentration inside the cell, they spontaneously drive a biophysical process called liquid phase separation, or LLPS. To visualize this, think of oil separating from water inside the cell, but with proteins. These viral components, the N protein, the P protein, and the L polymerase, spontaneously coalesce into dense, non-membrane-bound droplets.
And what's the advantage of this viral factory?
It offers two crucial functional advantages. First, it spatially concentrates all the necessary machinery for maximum RNA synthesis efficiency. Everything is right there. Second, and critical for its stealth, these factories sequester the viral RNA and replication components away from the host cell's immune sensors, particularly RIJ. This contributes significantly to its low-profile infection strategy. It helps it hide.
That makes perfect sense for a self-pathogen.
Right.
Okay, let's detail the 5 actors in this drama, focusing especially on their roles in antagonizing the host immune system.
Right. We can start with the most abundant, N, the nucleoprotein. Its primary role is obviously encapsulating the RNA, forming the template. It also regulates the switch from transcription to replication, which we'll get to. But on the immune front, N is a critical player in innate evasion by directly antagonizing RIG-I signaling.
So it stops the cell from even sensing the virus is there.
Pretty much, yeah. It prevents the cell from recognizing that viral RNA is present in the first place.
And the P, the phosphoprotein, is the immune system's nightmare, isn't it? Sounds like the real villain here.
It is the primary immune antagonist, definitely a strategic weapon. P is the necessary non-catalytic cofactor for L, linking it to the nRNA template. But its regulatory roles are multi-pronged. First, it exists as multiple isoforms that are critical for axonal transport getting around the neuron. Second, and most importantly, it targets the critical type I interferon, or IFN, pathway at multiple junctures. Yes, it doesn't just block one thing, it blocks 2 key steps. P protein inhibits IRF3 activation, which stops the cell nucleus from initiating IFN gene expression in the first place. And if IFN is successfully produced and signals to neighboring cells, P protein still wins by binding to phosphorylated STAT1, preventing that crucial signaling molecule from entering the nucleus.
Oh, it blocks the alarm in the response.
Exactly. By blocking STAT1, P effectively prevents the induction of hundreds of antiviral genes called ISGs, or interferon-stimulated genes. It's a systemic suppression mechanism. Very effective.
Okay, then we have M and G, the structural engineer.
Right. M, the matrix protein, as we discussed, is the orchestrator of assembly. It condenses the RNP and links it to the envelope. It also plays an internal regulatory role, actively inhibiting transcription and ensuring the virus shifts fully into packaging mode when assembly components are ready. It's sort of a quality control manager, too.
And G?
G, the glycoprotein, is the external spike and the primary gatekeeper. It mediates attachment and fusion, and it's the primary determinant of both pathogenicity and the virus's ability to invade the nervous system, what we call neuroinvasiveness. And as we said, since it's the sole target for neutralizing antibodies, its structure is paramount for vaccines.
Finally, the catalytic giant. Oh, the large protein.
L is the engine, the RNA-dependent RNA polymerase, RDRP, that drives all the RNA synthesis. It possesses all the required enzymatic activities. Polymerization, making the RNA chains. 5' mRNA capping, using its pRNTase domain to put a protective cap on the mRNA so the host cell can read it. And cap methylation. It's the most complex, most massive, and, due to that stop-start mechanism, the least abundant protein the virus requires.
This five-man team is incredibly efficient, it sounds like. Let's trace their life cycle now, starting with invasion and how the virus specifically targets nerve tissue. How does it get in?
Okay, so the process starts with the G protein-engaging host cell receptors. Given the virus's powerful neurotropism, it targets receptors that are heavily expressed on neurons and muscle cells, particularly at neuromuscular junctions.
Which receptors specifically?
Key entry points include the nicotinic acetylcholine receptor, NHEHR, the neuronal cell adhesion molecule, NCAM1, and the P75 neurotrophin receptor, NGFR. Receptor engagement triggers the virus to be basically swallowed by the cell through clathrin-mediated endocytosis, enclosing it in an endosomal vesicle.
So the cell essentially internalizes its own destruction. When does the RMP core actually escape that vesicle?
Once inside the endosome, host proton pumps activate, lowering the internal pH dramatically. This drop in pH acts as the critical acid trigger. It induces irreversible conformational changes in the G protein.
The shape change.
Exactly. This exposes a hidden, highly conserved fusion peptide, which then catalyzes the fusion of the viral envelope with the endosomal membrane itself. That final act of fusion releases the RNP core, the entire genetic machinery, into the cytoplasm. That is the uncoding event.
Okay, so once that RNP core is free, presumably sequestered within one of those Negri body factories, how does the virus manage the delicate internal switch from prioritizing making proteins to prioritizing making full-length genomes for new viruses?
Right, it starts with the transcription phase. The packaged LP complex immediately starts synthesizing complementary positive sense mRNAs for N, P, and G, and L, following that polar stop-start gradient we discussed earlier. The L protein adds the necessary 5' cap and 3' poly A tail to make those mRNAs readable by the host's ribosomes for protein production. The entire system stays in transcription mode, just cranking out proteins, until a crucial molecular threshold is met, the accumulation of newly synthesized N protein.
So N protein is the chemical switch. How exactly does the N protein flip the switch from transcription to replication? What does it physically do?
When N protein levels are low, the polymerase defaults to transcription, making mRNA. However, once the N protein reaches a high critical threshold concentration, it begins to stoichiometrically encapsulate the nascent positive sense RNA molecule as it's being made.
It coats the new RNA strand.
Exactly. This encapsulation event is the key. When the RNA is fully protected by N protein, it changes the template structure in such a way that the polymerase ignores the intergenic stop signals and remains processive, reading through the entire length of the genome without falling off. This uninterrupted process yields the full-length positive sense antigenome. That antigenome then serves as a template for synthesizing vast quantities of progeny negative sense genomes, all of which are immediately co-replicatively encapsulated by N protein, ready for packaging into new virions. That's the signal to shift fully into the replication phase.
That is incredibly elegant self-regulation. Okay, and the final movement is assembly and budding, which is where the M protein shines again, you said.
Yes. M protein is the master assembler and checkpoint regulator here. Its first job is condensing the newly replicated RMPs into their tightly coiled configuration, getting them ready for packaging. Next, it acts as a molecular bridge, linking that condensed RMP core to the cytoplasmic tails of the G protein trimers, which have already been trafficked to and inserted into the host cell membrane.
I recall the M protein also helps regulate the internal processes here, not just structure.
It does. It acts as a final checkpoint. The M protein actively inhibits transcription and stimulates replication, ensuring the cell prioritizes packaging over continued protein synthesis. This is a critical regulatory step, preventing the wasteful accumulation of unpackaged genetic material that could inadvertently trigger an immune response if left lying around.
Makes sense. And for the grand finale to actually leave the cell, how exactly does the cell membrane's own infrastructure get tricked into performing the final act of viral release? It doesn't just pop out, does it?
No, it performs the ultimate molecular hijacking. It steals the host's specialized machinery known as the endosomal sorting complex required for transport, or ESCRT machinery.
ESCRT?
Yes. The M protein contains a specific motif, a short sequence called a late budding domain, or L domain. This L domain recruits ESCRT components, such as a protein called TSG101, to the site of budding. Normally, the ESCRT system is involved in internal cellular membrane scission, like pinching off vesicles inside the cell. But the virus co-opts it to perform the final pinching off of the host cell membrane from the outside. This scission liberates the mature, bullet-shaped virion from the plasma membrane.
So without hijacking ESCRT,
The virus gets stuck. If you mutate that L domain on the M protein, the virus assembles at the membrane but remains tethered, unable to pinch off. It proves that the ESCRT co-option is absolutely vital for efficient egress and spread.
this efficient replication system leads directly to the virus's notorious lethality. Let's trace the journey from the initial bite to the brain. We know the incubation period can be prolonged, sometimes incredibly so.
It's highly variable weeks, months, sometimes even years, depending mostly on the site of inoculation. A bite closer to the brain means a shorter incubation, generally. During its initial phase, the virus replicates quite slowly in non-nervous tissues near the injury site, often striated muscle cells. The critical turning point is neuroinvasion. The virus finds its way into the peripheral nervous system, typically engaging receptors at the neuromuscular junctions, where nerves meet muscles.
Okay, once it's in the peripheral nervous system, it needs to be fast, right?
Yeah.
And it needs to bypass the massive defenses protecting the brain, like the blood-brain barrier.
It employs a brilliant strategy to achieve both speed and stealth simultaneously. It hijacked the host's fastest cellular transport system, involving a motor protein complex called dynein. Yes, dynein is responsible for transporting cargo backward along the neuron's axon towards the cell body. The virus uses this to perform fast retrograde axonal transport, moving centripetally, meaning towards the center along the axon toward the spinal cord and then ultimately the brain. This route is strategically essential because it completely bypasses the formidable, highly selective barrier known as the blood-brain barrier, which is designed specifically to exclude pathogens and large molecules from the CNS.
Clever, so it sneaks in the back door. What happens once it reaches the central nervous system?
Once it reaches the CNS, replication becomes extensive and systemic within the neurons. It spreads incredibly efficiently across synapses, the junctions between neurons, a process called transsynaptic transmission. It just hops from neuron to neuron. The final, crucial step for its own propagation is centrifugal spread. The virus reverses direction, moving along efferent nerves, those leading away from the brain, back out to the periphery. Critically, it infects the acinar cells of the salivary glands, ensuring it is shed in the saliva and ready for transmission to the next host via bite.
This leads us back to the pathogenicity paradox we touched on earlier. How does a virus that spreads so widely and causes such catastrophic neurological failure manage to do so with virtually no inflammation or obvious neuronal cell death? At least initially?
This is one of the most counterintuitive and, frankly, terrifying aspects of rabies. Post-mortem analysis shows widespread dissemination and high viral titers throughout the brain, yet surprisingly minimal inflammatory cell infiltration and little overt necrosis or cell death. The profound clinical dysfunction, the rage, the seizures, the hydrophobia is not caused by the virus mass-killing neurons, but by functional disruption. The virus is altering neuronal connectivity, ion channel function, neurotransmitter balance, essentially causing the nervous system to short-circuit and fail, while the cells themselves remain structurally intact, at least for a while.
And you mentioned an evolutionary explanation for why highly pathogenic or street viruses might actually replicate slower in cell culture than lab-attenuated strains. That seems backward.
Yes, it does seem backward, but it makes sense from the virus's perspective. The slow, stealthy replication rate is likely a calculated evolutionary trade-off. If the virus replicated too quickly and aggressively at the inoculation site, it would almost certainly trigger an immediate, potent local immune response that could clear the infection before it ever reached the sanctuary of the CNS. By maintaining a slow, low-profile replication rate initially, the virus prevents its own immune evasion mechanisms from being overwhelmed locally. It maximizes the critical window of opportunity necessary to achieve successful neuroinvasion before the adaptive immune system can fully react centrally.
The G protein seems to be the main dial that controls this delicate balance of stealth versus speed, based on what you said earlier.
Yeah.
Let's look at specific G protein virulence determinants again.
Absolutely. G protein is unquestionably the primary determinant of virulence.
Hmm.
Specific amino acid residues within it act as critical switches. Take, for example, the residue at position 333. The presence of an arginine at position 333 is profoundly correlated with a pathogenic phenotype in street viruses.
And changing it.
If scientists experimentally mutate that single residue to glutamine or glycine, the virus consistently becomes significantly attenuated. That single point mutation is often enough to basically break the virus's ability to cause fatal disease in animal models. Another fascinating example is the change from asparagine, ASN, at 194 to lysine, LYS. This single change increases pathogenicity dramatically. Mechanistically, sources show this change facilitates faster viral spread, more rapid internalization by the cell, and critically, it shifts the pH fusion threshold, meaning the virus can fuse with the endosomal membrane more easily and at a slightly higher pH, allowing for more efficient entry into the cytoplasm after being taken up by the cell.
And then there's the paradoxical, almost self-sabotaging mutation, where more G protein expression actually makes the virus less dangerous.
Right, the counterintuitive case involves a change from lysine to arginine at position 83. This mutation unexpectedly caused decreased pathogenicity, but only because it resulted in an unregulated increase in G protein expression levels.
How did that make it less pathogenic?
Well, that overabundance of G protein on the cell surface triggered more widespread apoptosis, or programmed cell death, in the infected neurons. It basically made the infected cells kill themselves too quickly. Furthermore, it seemed to increase the permeability of the blood-brain barrier. Essentially, the virus's overexpression of its surface protein accidentally turned the infection into a stronger signal for self-destruction and immune access, leading to attenuation.
Fascinating balance. Finally, let's revisit the specific strategies used for systematic immune evasion once the virus is operating inside the nervous system sanctuary.
Right. The virus operates within the neuron, which is already a somewhat immune-privileged site. Neurons naturally express low levels of MHC class I molecules, which are the flags that normally display internal peptides to alert cytotoxic T lymphocytes, CTLs, so they are inherently less visible to T-cell attack. However, the virus doesn't rely solely on the neuron's nature. The viral proteins P and N are the primary active suppressors, ensuring the rest of the cell's intrinsic defenses are shut down.
And P is the main culprit here.
We must emphasize the P protein's role again. It's the essential jamming signal for the interferon pathway, as we discussed. It blocks IRF3 activation, preventing the initial alarm signal from being sent out, and it simultaneously blocks STAT1 signaling, neutralizing the response even if an alarm does get through from neighboring cells. It's a complete two-part suppression. The N protein contributes as well by directly neutralizing Rabies I, which is the cell's primary initial sensor of viral RNA. Together, this multi-layered strategy of active suppression combined with the neuronal sanctuary ensures the virus can spread extensively before the body mounts any truly effective centralized immune response, often when it's already too late.
It's a dauntingly clever opponent, isn't it? But now, molecular science is being combined with powerful tracking technology to fight back. Let's talk about genomic epidemiology and how scientists are essentially reading the virus's genetic diary to trace its history and movements globally.
This is a truly transformative field, yes. The core principle is quite simple. As the viral genome replicates, it inevitably accumulates mutations, tiny random changes in its RNA sequence. These act like genetic footprints.
And we can follow these footprints.
Exactly. We use these footprints to trace the virus's entire evolutionary history and geographical spread. This involves two main techniques. Phylogenomics uses the generally consistent rate of mutation accumulation as a kind of molecular clock. This allows researchers to essentially rewind the tape and estimate the precise timing of evolutionary events. For example, when did a specific lineage emerge, or when did it jump between host species?
And the other technique.
That's phylogeography. This maps those phylogenetic relationships, the family tree, onto geographic locations. It allows us to reconstruct the virus's spatial history, identify the exact transmission routes it took, and even pinpoint geographical barriers, like mountains or oceans, that might be stopping its spread.
And this data reinforces the compartmentalization idea we talked about earlier. The virus tends to stick to a specific reservoir animal in a specific geography.
Exactly right. Generally, we see distinct transmission cycles confined largely to a single primary reservoir species within a given region. In North America, for example, we have distinct genetic variants sustained primarily in raccoons, separate ones in skunks, others in foxes, and various bat variants, and they rarely mix successfully between those cycles. However, globally, the picture is overwhelmingly dominated by the cosmopolitan clade, which is dog rabies. Domestic dogs remain the principal source for over 99% of human fatalities worldwide. That's the main target for global elimination efforts.
Our sources provide two fascinating case studies. They use phylogeography to show the power of both barriers and mobility in shaping rabies spread. Let's start with the Philippines.
Yes, the Philippines study beautifully demonstrates the power of geographical barriers. Researchers used whole genome sequencing and found that while all Philippine RABV belongs to one single subclade called SEA4, that subclade has clearly diverged into three distinct genetic lineages over time.
And the key finding.
Crucially, each of those three lineages is confined exclusively to one of the three main island groups, Luzon, Visayas, and Mindanao. The deep ocean channels between these islands act as highly effective barriers to dog movement, preventing genetic mixing between the island populations.
So what's the practical insight from that?
The insight is immediately actionable for public health. This genomic confirmation verifies that an island-by-island elimination strategy is biologically feasible. Because reintroduction of the virus from a neighboring major island group is highly unlikely, control efforts can be focused and sequentially implemented with a higher chance of success.
Okay, now contrast that island containment with the Arctic, where the environment seems immense, yet the virus is genetically homogeneous across huge distances. How does that work?
The Arctic rabies case is the perfect counterpoint to the island model. Viruses sampled across the vast circumpolar region from Greenland to Canada to Alaska all belong to a single, widely distributed lineage known as Arctic 3. There is essentially no significant geographic structuring across thousands upon thousands of kilometers.
And the reason is the host animal.
It's the mobility of the primary reservoir host, the Arctic fox. The Arctic fox is capable of traversing hundreds, even thousands of kilometers, often across sea ice, effectively connecting disparate populations across these vast distances. This constant movement genetically homogenizes the virus throughout the circumpolar landscape. The virus's genome basically tells the story of the host's incredible travel behavior.
Genomics also allows us to detect those critical, sometimes rare host shift events, right? When the virus jumps and establishes itself in a new species.
Precisely. That's a huge advantage of ongoing genomic surveillance. For instance, scientists in Canada used this approach to confirm that after a massive outbreak of the Arctic fox variant was largely controlled in foxes, the virus didn't completely disappear. Instead, it persisted by successfully establishing a new, sustained transmission cycle in skunks in the same region. This kind of high-resolution data allows us to catch these subtle but really significant evolutionary leaps, alerting public health officials that a new reservoir animal now needs to be targeted for vaccination and control measures.
Okay, so what are the tools and strategies driving the 0 by 30 goal, that ambitious target of eliminating human deaths from dog-mediated rabies by the year 2030?
The key is really integrating next-generation sequencing, or NGS, into routine surveillance programs. NGS allows public health labs to characterize the entire viral genome directly from routine clinical samples like brain tissue or saliva, very rapidly and relatively cheaply now. This high-resolution data is vital for distinguishing definitively between, say, an ongoing local outbreak getting worse versus a completely new importation of a distinct virus strain from another country or region. This allows control measures like vaccination campaigns or animal movement restrictions to be targeted much more precisely and effectively.
It helps with diagnosis, too.
Absolutely. Furthermore, untargeted metagenomic NGS, or MNGS, is becoming a diagnostic powerhouse. For patients presenting with atypical or confusing neurological symptoms where rabies might be suspected but isn't certain, MNGS can sequence all the genetic material in a sample host and pathogen alike. It can rapidly identify the infecting agent without needing prior knowledge or specific tests, preventing dangerous misdiagnoses. And to manage this explosion of genomic data globally, tools like the Method for Assignment, Definition, and Designation of Global Rabies Virus Lineages, or MDDoG, are being developed. These provide a standardized nomenclature system based on phylogeny, allowing researchers worldwide to speak the same genetic language when tracking specific viral lineages across borders.
Let's discuss the final frontier now, how this deep understanding of the virus, particularly the G protein structure, is revolutionizing our approach to treatment and prevention. This sounds like where the big breakthroughs are happening.
This really represents one of the biggest scientific advances in the rabies field in decades. For a long time, the precise atomic structure of the G protein trimer, the functional spike on the virus surface, was unstable and incredibly difficult to capture. But recent spectacular advances in cryo-EM, cryo-electron microscopy, have finally solved the structure in its native prefusion conformation.
What did that reveal?
It revealed previously hidden vulnerable epitopes, essentially the weak points or Achilles heels on the virus's surface that antibodies can target. This detailed structural knowledge is now driving rational vaccine design. Instead of just using whole inactivated virus like we've done for over a century, scientists can now engineer specific G protein antigens that are more potent, more stable, and crucially might offer much broader protection. The hope is to create vaccines that could shield us against those divergent Lyssaviruses in phylogroups the second to third that we talked about earlier.
And this structural clarity is also moving us towards safer, standardized treatments for post-exposure prophylaxis, replacing the older methods.
Absolutely. The high-resolution G protein blueprint accelerates the development and optimization of potent monoclonal antibody AM cocktails, such as one candidate called SYN023. These MABs are highly specific antibodies designed in the lab. These MAB cocktails can be mass-produced with high consistency, rigorously standardized for potency, and offer a much safer alternative to traditional rabies immune globulin, or RIG. RIG is derived from the plasma of vaccinated human or equine donors and is often scarce, expensive, and carries a small risk of adverse reactions, especially in developing countries where it's needed most. MABs bypass many of those issues.
Okay, now for the most provocative idea in our sources, challenging the centuries-old dogma that rabies is absolutely 100% fatal once clinical symptoms appear. Is that really being questioned now?
It is, and it challenges everything we have believed about rabies for millennia. The central dogma has always held that once the virus enters the CNS and symptoms begin, it's inevitably fatal that CNS entry is the definitive point of no return. However, some remarkable preclinical studies using aggressive high-dose combinations of these potent MAB cocktails have actually successfully rescued animals, like mice or non-human primates in experiments, even when the treatment was initiated late, after the virus had demonstrably reached the central nervous system and neurological signs were beginning to appear.
Wow, so what does that imply?
It strongly suggests that the true point of no return might not be CNS entry itself, but perhaps the later onset of widespread, irreversible neuronal damage. If we can flood the CNS with potent neutralizing antibodies early enough in the symptomatic phase to shut down viral replication before that permanent functional damage occurs across critical brain regions, we might actually be able to stabilize the patient and allow for recovery. The future of potential rabies treatment, not just prevention, is now shifting toward exploring combinations of potent antiviral MABs with neuroprotective or neuroregenerative agents, trying to treat the viral infection while simultaneously protecting or even repairing the damaged nervous system. It's a huge paradigm shift.
And for prevention, particularly in endemic regions, new vaccine platforms sound like they're finally solving some long-standing logistical problems.
Yes, that's another exciting area. New vaccine platforms, such as recombinant viral vectors like the chimpanzee adenovirus-based vaccine, ChAdOx2 RABG, similar technology to some COVID vaccines, promise solutions specifically tailored for regions with limited resources and infrastructure. These new platforms have the potential to be single-dose, highly thermostable, not requiring constant refrigeration, and much more cost-effective to produce at scale. This could make mass prophylactic vaccination campaigns in humans and, critically, high-volume veterinary vaccination in dog populations truly feasible in the remote endemic areas where the need is greatest and where the cold chain is hardest to maintain.
This deep dive into the Lyssavirus has been, well, truly remarkable. We've seen that rabies is not just a disease of historical horror, but actually a marvel of evolutionary efficiency. Just 5 genes orchestrating a systematic, stealth-dependent life cycle, relying heavily on the P and N proteins to dismantle the immune system from within the neuron sanctuary.
And the fight back is becoming equally sophisticated, isn't it? The convergence of molecular science finally solving the G protein structure using cryo-EM and high-resolution genomic tracking via NGS and systems like the MAD-DOG nomenclature provides precise, actionable intelligence. This dual-front assault, combining better tools with better tracking, means the 0 by 30 goal of eliminating dog-mediated human rabies seems increasingly attainable. It's driven by both the potential for improved mass vaccination and the incredibly precise maps of viral movement provided by phylogeography.
So here's the final thought for you, our listener. The rabies virus has been defined by its near 100% lethality for millennia, an absolute death sentence once symptoms appear. But if this new research continues to show that the irreversible functional tipping point actually comes later than we previously believed, what impact could the ability to potentially treat established CNS infections actually have on global disease elimination strategies? And maybe more profoundly, how might that fundamentally change our relationship with this ancient pathogen, moving us, perhaps, from fearing an inevitable death sentence to cautiously anticipating a potential post-symptomatic cure in the future?